Previously, we've learned about the idea of spontaneous generation, which says that life can emerge from non-living materials. The idea has a long history, going back a few thousand years, and one example would be exposed meat spontaneously generating into worms. It wasn't until the mid-1600s that Francisco Redi tested this idea with the experiments you see here. And you'll recall he made careful observations and came up with an alternative hypothesis about the origin of the worms. He thought that flies were landing on the meat, laying eggs, and the eggs turned into the worms, and the worms ultimately into flies. So the worms were part of the life cycle of flies. Well, to test these two hypotheses, he set up this experiment. And again, you'll recall that this being the experimental condition because the variable fly access, or the eggs landing on the meat, right, or being put on the meat, this jar has the variable, so this would be the experimental condition. He set up a control condition, where he blocked access to the meat. The flies could not get to the meat. Sure enough, there were no worms found in this condition. However, believers in spontaneous generation argued that perhaps spontaneous generation requires fresh air. After all, they thought that nature had a vital force, some kind of productive power or energy that was responsible for spontaneous generation. And if it was carried by the air, none of that force could get into this chamber because of the lid here. So Redi fashioned another control condition where he, instead of a lid, put cloth over the jar. This allowed air, but it blocked the flies. Sure enough, he found no worms here. So from these experimental results, Redi concluded that spontaneous generation is not happening in the case of the meat here. Rather, the worms that you might find on meat come from fly eggs that were placed on the meat. Even so, many people continued to believe that spontaneous generation was common in nature. So while meat might not spontaneously generate into worms, what about these things? Now, what you're looking at are pictures of bacteria. These are the smallest cells on Earth, and they were only discovered after the invention of microscopes. So in the 1600s and 1700s and 1800s, when microscopes became more widely used, scientists discovered these smallest of living things. Single-celled organisms, they are bacteria. And so while people thought that, okay, Redi convinced everybody that meat doesn't spontaneously generate into worms, nevertheless, many argued that these living things could spontaneously generate from non-living materials or non-living fluids. So in this lesson, we're going to look at some of the early research on spontaneous generation with respect to bacteria. So the first set of experiments we'll look at were performed by John Needham in the 1700s, and he was a vitalist. And again, the vitalists were people who believed that nature had this kind of vital force that had the power to create life from non-living materials. Needham is going to do the following experiment here, and he's going to argue that his results supports the idea of spontaneous generation. Let's see what he did. So he takes some broth, now meat broth. Now this is the sort of juice left over after you're cooking some meat, and it seemed to be a favorable medium with for which bacteria could grow. And so he's going to take a flask full of this broth here, and he's going to boil it. Now he wants to take a sample, then he's going to check it, put it under the microscope to make sure there's no bacteria. So this boiling process here is intended to kill any bacteria that might be present in the broth. So if you're going to do experiments on spontaneous generation, right, you have to start with non-living stuff, in this case non-living broth. So sure enough, he checks some samples from his jar, and he doesn't find any bacteria. He puts a cork in his flask and waits some days. He then takes the cork off and checks a few more samples, and voila, he found bacteria. So from Needham's point of view, he has killed whatever bacteria were in there to begin with, and he confirmed that with his checking procedure here. A few days later, bacteria are found. Well, where did they come from? Spontaneous generation. Now, at this point, we're going to introduce a rival to Needham, and his name is Lazarus Spallanzani, also living in the same time period. Spallanzani is going to question Needham's interpretation. He's going to be critical. Spallanzani is not a believer in vitalism. He's not a vitalist. He doesn't think that nature has this this vital power or force to spontaneously generate living things. And Spallanzani is going to look at this experiment, and he's going to have several criticisms. Now, you might take a moment to pause and think to yourself, well, how might we criticize Needham's interpretation? So here we see his procedure. He concludes that the bacteria must have spontaneously generated, but Spallanzani is going to argue that there are alternative explanations for the bacteria at this point. So we could pause the video here if you want to think up on your own what are some possible criticisms. Okay, so here goes Spallanzani going to be criticizing Needham. First of all, the boiling procedure has to be a procedure that kills all the bacteria. So one thing Spallanzani can say here is that Needham did not, in fact, boil long enough to kill all the bacteria. Well, why didn't Needham find any bacteria when he was checking? Well, perhaps just that particular sample had no bacteria, but there might have been bacteria at other locations in the fluid. Right? And so that's another kind of criticism, that the checking procedure here was not sufficient to identify that there really were bacteria in the broth. And a third criticism might be the cork here. Perhaps the corking procedure either introduced bacteria into the vessel or it allowed bacteria through the seal, the cork seal, into the vessel. So on a variety of counts, Spallanzani is going to criticize Needham's interpretation. Now, Spallanzani, after criticizing Needham's interpretation of Needham's experiment, Spallanzani is going to do a series of experiments of his own, trying to improve upon the techniques of John Needham. So he's got 4 flasks here, and they're going to go through different procedures. Flask number 1. Notice he's got some broth here in a flask, and he's going to boil it. But notice this flask does not have a cork. In fact, what Spallanzani is going to do is he's going to melt the glass shut here. So the boiling now is the broth will be boiled, but the glass will be melted shut. So you don't have the problem of the cork seal. He boils for 10 minutes, and then a couple days later, going to break off the top of the flask, check, and he finds bacteria. Flask number 2, also sealed with melted glass here, boiled for 1 hour. And leave it sit for a couple days, break it open, no bacteria. Then he had 2 other kinds of flasks. Flask 3 here, like Needham, he had a flask with broth and then corked here, boiling for 10 minutes, wait a few days, did find bacteria. And then another one, also corked, boiled for 1 hour, wait a few days, also had bacteria. So notice what Spallanzani is doing here. Flask 3 and 4, it's like he's trying to reproduce the conditions of Needham's experiments. That's good scientific practice, to try and replicate, they say, replicate somebody's findings. But flask 1 and 2 are a new technique. Instead of using cork, he's melting the glass shut to prevent any contamination from outside. So let's see how Spallanzani interpreted his results. For flask number 1, he found bacteria. Now recall he boiled for 10 minutes, although it was sealed shut, it was melted shut. But Spallanzani would argue that the 10-minute boiling process didn't kill all of the original bacteria, and that's why he found bacteria later when he checked after a few days. For flask number 2, the 1-hour boil, he argued, did kill all of the bacteria. However, because no bacteria could get in because it was melted shut, and because there is no spontaneous generation, this is why there was no bacteria found in this flask. Notice, Spallanzani here is going to interpret this second flask as evidence that spontaneous generation does not happen. After all, why didn't it happen? We found no bacteria here. There was a broth here, and if broth can spontaneously generate into bacteria, it should have happened here. In flask 3, a 10-minute boil but with cork, here Spallanzani argued that 10-minute boil didn't kill all of the bacteria, just as in flask 1. And then in flask 4, with a 1-hour boil, in fact, he found bacteria. But again, like this one here, the 1-hour boil may have killed all of the original bacteria, but evidently, said Spallanzani, bacteria could get in from the outside through this cork seal. But it could not when the glass was melted shut. So Spallanzani is going to interpret his overall results in favor of the idea that spontaneous generation does not happen. Rather, bacteria come from other bacteria. However, as we'll see here, John Needham is going to have a different set of interpretations for Spallanzani's experiment. And this is characteristic of the history of science. When we have rival scientists with rival ideas about the world, they will interpret each other's experiments in different ways, usually in favor of their own set of hypotheses. So let's see what Needham is going to say about Spallanzani's experiment. So for flask number 1, Needham is going to argue, well, this result is consistent with spontaneous generation. After all, so you might be boiling here for 10 minutes, but that 10-minute boil was not enough to destroy all the vital force there, and so you could get spontaneous generation inside the flask. For flask number 2, the reason why you get no bacteria is because the 1-hour boil did destroy the vital force in the air in this vessel, and so it could not mix with the broth to trigger spontaneous generation. So no bacteria in flask number 2, but Needham has his own explanation for that. Flask number 3, 10-minute boil, may not destroy all of the vital force, and perhaps some could still get in through the cork seals, so we have the conditions for spontaneous generation. And same with flask number 4. The boiling might have destroyed the vital force in the air here temporarily, but more might have been able to get in through the cork seal and to allow for spontaneous generation. So we see we have a problem here, that both Needham and Spallanzani are interpreting the same experimental results in different ways. Ultimately, the problem has to do with this idea that spontaneous generation is dependent upon this kind of vital force, or active principle, as Needham called it, which is carried by the air. And so when Spallanzani came along and melted his flasks shut, well, this set up a situation where boiling might destroy whatever vital force was in the air here, and then not allow any new, fresh vital force in. So the believers in spontaneous generation could maintain their belief in spite of Spallanzani's results. What was needed was for somebody to come along and do an experiment like what Reddy did for the meat. You'll recall believers in spontaneous generation again said that air might be required. Well, Reddy had a control condition that allowed air. And the same kind of thing is going to be needed for the case of the bacteria. Somebody will have to come along and set up a condition, some flasks where we can boil the broth, kill whatever's bacteria is there, but still allow fresh air into the flask without carrying any contaminating bacteria from outside. And the person to do those experiments has a name, Louis Pasteur, and we'll see his experiments in the next lesson.